Hi, this is Miss Litton, and this is a Monday afternoon, and there's a bunch of AP biologists. Come on in, just ready to review. Say hi. Hi. Okay. All right. I'm going to tell you for sure some things. Number one, this is chapter 10, which is about Meiosis. But I will ask you questions to see if you can differentiate between meiosis and mitosis. So you better keep it. So if I say sister chromatids separate, does that happen in both meiosis and mitosis?
No.
Sister chromatids separate. Does that happen in mitosis and meiosis? When? Tell me in mitosis when it happens. Anaphase. And when does it happen in mitosis? Anaphase. And then in meiosis it would be anaphase 2. Because what happens in anaphase 1? Homologous pairs. Do you see how I can check to see if you know it? And that would be one of those math questions. What does that mean? Okay. Yeah, I say multiple answers possible, but yeah, mark all. It has to be an elective question. All right. Never mind. All right. So the formation of gametes is through meiosis. What is the same about meiosis and mitosis? Both times the DNA what? Replicates. But in meiosis, you have to do not one division, but two. The first division is focusing on separating the homologous, and the second division you're separating the sister chromatids. Do not feel compelled to say it faster than I can ask the question. That way we can allow everybody to think about the answer. Okay. Do we do alternation of generations?
No.
No. Who does alternation of generations? Plants. What are the two generations? Sporophyte and the gametophyte. Gametophytes always make gametes. Sporophytes always make spores. Gametophytes always make gametes. Sporophytes always make spores. Right? Okay. We have the only haploid structures. Every single thing in your body is diploid except what? Except your skin cells? Sex cells. Sex cells, everything else is diploid. If I ask you about any other cell in your body, if I ask you about the islets of Langerhans in your pancreas, it's going to be those are diploid because they're in your pancreas. The only ones that can be haploid are what?
Sperm or egg.
Sperm or egg. What's another name for sperm or egg? Gametes. Good. All right, just check it. When we talk about homologous pairs are the same size, the same shape, and they code for the same characteristics. Could you have variations in how they code for that characteristic? Yes. And those variations are called alleles. They're called alleles. Those are called alleles. Okay, so for instance, this is right here, this gene is the same size, same shape, same characteristics for the homologous pair, but this alternative forms of a gene, white flower versus purple flower, those are alleles. Blue eyes versus brown eyes, tall versus short, round versus wrinkled, right? All of those are alleles. And something that sets apart meiosis is the opportunity for variation. So when you look at these cute puppies, you should come to mind that this is going to, the reason why there's variations in the puppies, there are 3 reasons why. That would be an excellent essay question. Okay, so in your mind, come up with the 3 reasons why there are variations in these puppies. Would you start off something about telling me how they are sexually reproducing organisms? Would you start off by telling me that they would undergo the process of meiosis to form haploid gamete cells? Would you tell me in the process of meiosis that there are 2 areas for variation? One, and the first one is what? Crossing over, which occurs when? And what is crossing over? Homologous pairs do synapses, and you exchange with what? Sister or non-sisters? Non-sisters to get variety. If you exchange with sisters, there's no variety with that, right? What is it called if you do crossing over with not your homologous pair? Translocation. Think about it. Translocation. I am changing my location. I used to be on chromosome 7. Now I'm on chromosome 17 because I did synapses with the wrong one. Translocation. crossing over not with a member, a homologous pair. What is it called when you take away from a chromosome through the process of crossing over? Deletion. And if you get a little something, something extra? Addition. So deletion and addition. If somebody's getting deleted, when somebody's getting deleted, somebody else is getting added too, right? Because it's going somewhere. I mean, unless it just, goes away. But let's talk about inversion. Inversion is when you do crossing over and a section of that chromosome gets what? Reversed or flipped. Yeah, flipped over. Does that make sense? So when we talked about that at the very end of this chapter, it was all referring to what could happen during meiosis, and that's specifically during crossing over. Okay, then where's the second place for variation? Independence. Right. And so the homologous pairs line up when? Metaphase 1 of meiosis. And the way they line up, each homologous pair can line up independently of every other homologous pair, so long as the homologous pairs are in the middle. Anaphase 1, right? You separate the homologous pairs. Anaphase 2, you separate the sister chromatids. What is it called if you have failure to separate? Non-disjunction. What is the most common form of non-disjunction on the autosomes, the non-sex chromosomes? Down syndrome, which is known as trisomy 21. Trisomy 21. Good. When we talked about Turner syndrome, which is a single X chromosome, or Klinefelter's, which is XXY, that would also be a result of non-disjunction, right? One got an extra X, one got one less X or a Y. Okay, that is also a form of non-disjunction, but in sex chromosomes. Does that make sense? Okay, so that's how we have variety. You have crossing over during prophase 1. You have independent assortment during metaphase 1. Let's check these questions right here for starters. I have some other review questions in here as well. I was trying to actually say the words. Let's go in as our favorite Monday afternoon activity. Yes? Wait, let me pause. Okay, go ahead, ask your question. Guys, you let people ask their questions. Shh, go.
For a person, if you have any extra chromosomes, would that be non-disjunction?
It would be a result of non-disjunction because you got extra.
Okay.
Yes. Eating, crying, and stressing. Band practice, a nap. Unit 3 partner test. Eating. Don't die. Don't die. Life's precious. Life's precious. How come we don't write those things down? Life's precious. I love you. But can we write those things? It's in here. Okay. I'm going to pause for a minute. All right, let's check it out. Okay, guys, it will facilitate our time together. Gentlemen, it'll facilitate our time together if we can talk through this rapidly. Okay, synapsis and then crossing over. Map occurs during synapsis is when the homologous pairs come together. Crossing over is then when they exchange, after they've synapsed together. Will it occur during prophase 1?
Yeah.
Yes. Right? It's going to happen before they're totally lined up. Will it increase variety? Yes. That makes sense. Does it occur between sister chromatids? No. How could we change this so it would be right? What would we have to do?
Add non-sister.
Non-sister. Occurs during metaphase 1? No. It only occurs during prophase 1. That means you cannot do it in meiosis 2. If you haven't done it in meiosis 1, it's not happening. Does that make sense? Okay. Occurs between non-sister. We got that. Okay, and then those are just other stages. Ask your bio buddy, are you in the Shadowlands on that question at all? Okay. Come back to me. Guys, the longer I have to wait, remember you don't have to be here. You can go home. Okay. Independent assortment, does it occur during prophase 1?
No.
When does it occur? Metaphase 1. Can it happen in metaphase 2? No, it's referring to homologous pairs being on the chromosome. In the middle. Increases variety, yes. It doesn't occur then. It doesn't have anything to do with chromatids. It does occur between homologous pairs because it's one homologous pair to another homologous pair. And occurs at yes, and that's it. Ask your Bible, are you okay on that one? And fertilization also increases variety because these were the three. Crossing over, independent assortment, and fertilization because you don't know what sperm is going to fertilize what egg. It definitely doesn't occur during asexual. It doesn't occur during any of those. Is between gametes, yes. Occurs in sexual reproduction, yes. Okay, shadowland, sunshine on any of that? Okay, let's not get distracted. Let's go through mitosis ones, and then we'll go through meiosis ones. Okay, then we'll go on to the next part here. So, reviewing mitosis, the first step is what? Do it with me, quickly, let's go. First step is what? Oh, that's the cell cycle. First step of mitosis is prophase. And we don't say ones or twos after it, because there's only a single division, okay? The chromosomes become visible, the nuclear envelope and the nucleolus disappear, radiating microtubules, asters. They meet in the middle at metaphase, single file, away at anaphase, interchromatid, to nuclei at telophase. Cytokinesis, pinching off cleavage furrow in animal cells, or what?
Cell plate will become cell
Cell
wall.
plate will become a cell wall. In what?
Animal.
Okay, now let's do meiosis. Okay, so now let's do meiosis. The chromosomes become visible. That part's true, right? The nuclear envelope and the nucleolus disappear. What stage are we in, by the way? Okay. Disappear. Radiating microtubules, asters. What else occurs that's unique to prophase 1? Synapsis. And then what happens? Crossing over. Okay? Then what happens? What's the next stage? Metaphase 1, right? They meet in the middle at metaphase. What am I showing now? Independent assortment. What is this increasing? Variety. What else increased it? Good. They meet in the middle at metaphase 1. Thank you. Away at anaphase 1. Who went apart? Okay. To nuclei at telophase. Now, telophase 1, but now it's also haploid cytokinesis. Why is it a haploid cell now?
Because it doesn't have
Right, it doesn't have the homologous pairs. Okay, so show me prophase 2. Show me metaphase 2. It's hard, good, Amelia. And then what comes next? Anaphase 2. Hatch it. And then telophase 2, cytokinesis. And how many cells do we have? 4. 1, 2, 3, 4. If I'm doing what in humans? Yes, spermatogenesis will make you 4 haploid sperm, right? What is it called in females? What's the process? Oogenesis. And we will make, I have to be careful. 1 egg and what? 3 polar bodies. 1 egg, 3 polar bodies. Okay? We don't even do this. Was done before we were born. We don't do this until the sperm shows up. Then we're like, okay, then we'll do it. And that's why, because this doesn't happen until the sperm shows up, if you have a 40-year-old egg with hello sperm, instead of hello sperm, sorry, then sometimes these fail to separate, and what is that called? Non-disjunction. Good, perfect. Oh, life's lovely. All right. We talked about life cycles. We talked about our organisms. We're always diploid. Animals are always diploid. The only ones that are haploid are sperm or egg. We talked about alternation of generations. We're going to hit this harder when we get into plants. But for right now, you just need to know that I will highlight what is diploid. Right? The zygote's going to be diploid. Everything here, right? Here, the sporophyte's diploid, but then everything else is haploid. Right? The sporophyte always makes spores. Gametophytes always make gametes. So the sporophyte is diploid, but it makes its spores by doing meiosis. So the spores, when you have those spores, they are haploid. The haploid spores grow up to be the haploid gametophyte. The haploid gametophyte does mitosis to make haploid gametes. The gametes get together through what? Fertilization to make a diploid zygote. Yeah. As plants moved on to land, the sporophyte gained ascendancy. It became more and more dominant, and the gametophyte became less, and then is dependent upon the diploid sporophyte. Okay, and then we've already compared and contrast mitosis and meiosis. What is this?
Karyotype.
A karyotype, which is a picture of your chromosomes, and that allows us to see if we have the correct number of homologous pairs, or see if we are male or female. Yes? Okay, here we go. All right, let's check it out. Now, why would we want to say I'd rather happen in meiosis 2?
Because meiosis 2, you'll have 2 damaged cells, whereas meiosis 1, you'll have 4 damaged cells.
Exactly. Exactly. Nice, nice. Okay, so then we just talked about a yellow highlighter. Polyploidy. What does poly mean? Yeah, so we know diploid, haploid. This is polyploid. Okay? Extra sets of chromosomes. Incorrect. Aneuploidy, just right. You are just right. Euploidy. And I don't know how to do that. There's nothing. What? Yeah. You are just right, euploidy. We also talked about, guys, I can't get this. In a nucleus, not all the DNA is active at the same time. By active, I mean getting transcribed into mRNA and translated into proteins. So some of that DNA coils up, and that's called heterochromatin. Bar bodies are an example of heterochromatin. We have a double dose of X chromosomes in females, so one of them will become that heterochromatin, that bar body. We looked at pictures of that. We did some sentences. I think we can skip that for right now. We talked, if we have time at the end, you can do it. We talked about Down syndrome being a form of non-disjunction, and the most common autosomal trisomy. Then we also talked about a monosomy and Turner syndrome and a sex chromosome, and a trisomy for an extra chromosome for Klinefelter's XXY. And then we just finished up by talking about additions and deletions and inversions and translocations. And then I have some questions for you here. I'm going to pause you because there are 8 questions. All right, let's check. Okay, guys, it would help. You can't block the door right there, sweetheart. It would help, there's plenty of chairs. It would help if we went through just the ones that missed and we identify the correct stage for it. So on question number 1, sister chromatids separate. Somebody said E, which is anaphase 1. What separates an anaphase 1?
Homologous pairs.
Homologous pairs. Okay, number 2. Yay! Crossing over only occurs in prophase 1. That's not something where you like go, I don't understand. It's just like, this is my left hand and this is my right hand. Crossing over occurs during prophase 1. I don't know what else to tell you. This is a Chromebook. This is a water bottle. Synapsis and crossing over occurs during prophase 1. Okay. Name the stage where there are 4 daughter cells that are haploid. Is that what I said? Yeah, so it'd have to be telophase 2. Okay, in anaphase 2, you're separating. You haven't formed the nuclei yet to say that you have those daughter cells. Number 5, single chromosomes are being formed. Nuclear envelope may be breaking down again. Single chromosomes are being formed. Nuclear envelope breaking down again. So what stage does the nuclear envelope break down? Prophase. So then you just have to decide, is it prophase 1 or prophase 2? And since I said again, you're going to go with prophase 2. Plus you don't have any homologous pairs. Okay, number 6, name the stage of meiosis where homologous pairs are separating. And that's just going to be always anaphase 1, because that's the only time you have it. Name the stage of meiosis where homologous pairs are in the middle, and then you would say metaphase 1. Okay, and then we're just going to add a layer in here as I can work my way backwards with these questions. Name the stage of meiosis where single chromosomes are in the middle. You would say metaphase 2. Tell me a stage of mitosis where there are single chromosomes in the middle. Metaphase, right? Okay, let's do name the stage of mitosis where homologous pairs are in the middle. No stage. No stage. Name the stage where homologous pairs are separating. No stage in mitosis. Number 5, name the stage of mitosis where single chromosomes are being formed, nuclear envelope may be breaking down. Prophase. But not again. Once I put again on there, it throws it out, yeah. 4 haploid cells are formed. No stage. Crossing over occurs. No stage. Nuclear membrane rebuilds for the first time, 2 cells total. Telophase. And you would just say telophase, right? And sister chromatids separate. Anaphase. Perfect. I don't remember what this is, but you can try it or we can do it together. You want to try it? Yeah. Okay. Did it play yet? There you go.
I'm not going to read them again.
Can anybody help me with deletion so I can do this for people at home? What one would go with deletion? Missing. Let's see. Yay, we're safe. What one goes with XO? Turners. Yay. What one goes with trisomy 21? Yes. What one goes with translocation? Perfect. What one goes with XXY? Mm-hmm. What one goes with inversion? Flipped. What about duplication? Extra. Good. Did you get it all right?
Yes.
No, I don't want to do that. Okay, thank you. All right. Another thing you probably want to be able to do is if I said, please draw for me a picture of mitosis, a cell in mitosis with mitosis, metaphase, diploid number 2. Diploid number 2. So 2n equals 2. Could you draw that? Okay, go for it. All right, so when I say 2n equals 2, how many chromosomes are there total? 2n equals 2. How many chromosomes are there? 2n equals 2. 2 chromosomes. How many are there going to be in the gametes? 1. Exactly. Because 1 times 2 is 2. Okay, so in metaphase, those two chromosomes will be lined up like this, yes? But if I'm looking at metaphase 1 in meiosis, they'll be lined up like this, yes? And in meiosis 2, they'll be single cells like this. So do you see, I could ask because it would happen in 2 cells, right? Because this one, you're going to separate the homologous pair, right? When you do this, so you'll end up with 2 cells that look like this. And here are those 2 cells ready to undergo metaphase 2. Okay, right here, these are going to end up like this. Yes?
Yes.
And I start off with 2 cells, and I'm going to end up with 2 cells. So you want to try to do that, you know, change it around. Go, you know, you want to do even numbers, you know, but I would do the same strategy, and I do it with 2n equals 4. Could I do that across the end? Do I know what that means? What would a 2n equals 4? What would a 2n equals 4 in metaphases across? How about in anaphases across? Try that and see if you agree with your bio buddies, and that will be a good way to indicate whether I know you know the stages and what occurs in each. Okay? Identify at any point where there could be an opportunity for a variety in any one of those stages. That would be important to know as well. Okay? Questions, concerns? Okay, you're super smart. Don't forget to do your highly suggested reading and thinking. And know that I love you, and if you get hungry, have a piece of toast, because toast is always yummy. Maybe put a little peanut butter and honey on it, you know.